Welcome back, guys. So today I am going to be covering how genes and genomes evolve. This topic kind of is going to touch both with the molecular and cellular biology side and also with the genetics group. So it's kind of going to be covering a little bit of both of those things. So what exactly does it mean when I'm talking about genes and genomes and how they evolve? So we're going to talk about how we generate genetic variation in organisms that are here. And then we're going to talk about how different organisms are related to each other and how we can reconstruct the relationships between one organism and another and classify them through a family tree. And then we will talk about major ways in which genes evolve, whether it be environmental factors as well as actual mobile genetic elements and viruses that can also act as those agents to move genomes around to create that genetic variation that leads to evolution. And finally, we're going to examine a little bit of the human genome and look at how different, how variable it is within humans as well as across the organisms, across the life tree. So why do we need to talk about it? So at the end, all humans essentially have the same exact genome, right? At the end of the day, it's a very small differences if you look at it at the at the nucleic acid level, at the sequence of the genome. You'll see some variations here and there, but overall, it's a really large genome. It has lots and lots of, billion, billions of nucleotides, and there are very few differences. A very small percentage of that is going to be different between different individuals. Yet, when you look at the human population, they have extremely different phenotypically, right? No two humans are going to be exactly the same unless you're looking at identical twins. Even then, there are going to be differences between them at some level. So, what is it exactly that makes just a small, tiny amount of differences give us this completely different, completely individual person when we are looking at the humans? Um, So we are going to talk about how those specific mutations and how those specific changes allow for such variability to occur, right? Essentially, all these changes that we are looking at, they are mutations, right? All those different variations. Initially, there was just one flavor of this gene, and then through environmental factors, genetic factors, a combination, you started introducing single mutations here and there. And those single changes or mutations led to, over time, slightly different flavors or versions of the same genes. And that's what we call alleles. And we've talked about that before as well. Now, some alleles are going to be no different than our typical phenotypically, because the alleles still lead to the same protein product at the end of the day. And that's because of the degenerate nature um, of the code. And that's something we have talked about, how different codons can still lead to the same amino acid as the product, because that third base pair can have some variability without having an end point effect. However, in other cases, you may actually end up with a different protein product at the end of the day, either because one amino acid is different or multiple amino acids are different, or there is a truncation or anything unique like that. Now, your personal combination of alleles is not going to be exactly the same as your siblings or another person's. Everyone's going to have a slightly different, or sometimes majorly different, combination of these alleles. You may match with some of them, and others are going to be different between parents, between siblings. And that variability is what is going to give you those different phenotypes then at the end of the day, leading to the differences in the hair color or the eye color or the way you behave, what kind of health benefits you have or susceptibilities you have, are all going to be dependent on these alleles at some level. So let's talk about how we generate these alleles. How is it that we are going to generate these variations in our genome? What are different ways that they can be done? So there are many, many different ways that we can do them, both environmental and genetic. We're going to focus more on the genetic side of it for now. So genes and genomes in general can be altered by many different mechanisms. This includes direct mutations to the coding sequence of your gene, right? So this is where looking at just that coding sequence of the genome of a particular gene, let's say for brown eye or blue eyes or anything else, there is a mutation somewhere in there in one nucleotide or multiple nucleotide, maybe a portion gets deleted or added, or just a point mutation happens that leads to a difference in the protein product in that particular codon, so that a different amino acid is there that leads to a different isoform or different shape or a different protein product at the end of the day, and that would be then your altered genome. You could also get these genomes or gene expression to change by not necessarily having a mutation in the coding sequence, not in the actual gene itself, but rather its regulatory DNA. So for most of our genes, you have very, very strict regulation of their expression levels so that they're not active all the time, or they're active only under certain conditions, or when certain stimuli are given, or certain time period is there, certain developmental time point is there, whatever the case may be. And those regulations are maintained by regulatory sequences within the DNA at many levels. Now, mutations in some of those regulatory DNA sequences would have an impact on the expression level of the related genes that are affected by those regulatory DNA. So if we somehow change that regulatory DNA so that now the regulatory protein cannot bind to it and cannot activate it or inactivate it, then it's going to disrupt the normal expression pattern of that particular gene and lead to a mutation in that way. You can also have gene duplications and divergence, especially if your DNA sequence is small, maybe for that particular gene, and is very repetitive. It can lead to duplications and multiple copies of the genes being found in multiple places, maybe even. And over time, they can diverge from each other and become different. So gene duplications and changes, subsequent changes in them can be another way that mutations are introduced and evolutionary changes are introduced into your genome. Another one is exon shuffling. So if you remember from before, genes, if you look at just the DNA sequence for a particular gene, it is going to contain introns and exons. Now, those introns and exons are going to be together what gets copied at the beginning of the time, right? And then you have the introns removed and all the exons joined together, spliced together to make your final protein, the final RNA, the messenger RNA that's going to then be translated into your protein product. Now, exon shuffling can be of two ways, right? So the intron sequences get spliced out, and your mature RNA is then made of all the exons spliced together. But those exons can be spliced in different orientation, leading to different gene products, right? Different protein products at the end of the day. And they don't have to be all from the same gene either. You can even have two different genes and their sequences and their exons getting spliced together, leading to exon shuffling, where the protein product is now made of some of protein A, some of gene A and some of gene B exons mixed together. And that's another way that these can form. And here's an example where you're seeing something similar, where there are two different genes, gene A and B, both have one intron, but when those introns are spliced out, when they're put back together, you've ended up with a mix and match where there are exons from both genes represented in both sides. That's another thing that can happen that can lead to a completely different protein product, obviously, because now each of those genes have different different domains in their final protein product. So, for example, let's say gene A had a nuclear location system, right? A nucleus location system, so NLS, or had a DNA binding domain that was mixed with its whatever other function it performs. Once it's spliced in this way, maybe it loses that nuclear location signal, and that gets moved to gene B. While now gene A product cannot go to the nucleus to have its transcription factor activity because it doesn't have that location system signal anymore. And gene B, which does something completely different, is now getting translocated into the nucleus because it has the NLS. So that is another way that you can disrupt the normal function of the gene, normal location of the gene as well, or the final product of the gene. Another way that you can disrupt genes are through transposition. So we've talked about jumping genes and transposable or mobile genetic elements. Mobile genetic elements can force or can take parts of genes or complete sequences, parts of the sequences or complete gene even, with it as they are spliced and moved to a different location, as they transpose to a different place. They can also themselves insert into the regulatory DNA sequence, which can lead to changes in the regulation of that particular gene, because now the regulatory sequence is disrupted with the existence of this mobile genetic element, and that's going to cause it not to bind to its normal regulatory proteins and not to have the function that it has to perform. And finally, the last one is horizontal or lateral gene transfers, where you have genes that are moving from one organism into another. And there are some cases that does happen, and we'll talk a little bit about that soon as well. Oops, sorry. So how do these happen, or many of these happen? Well, in sexually reproducing organisms, the only changes that matter, the only mutations that matter, are changes to the germline, right, to the actual germ cells, because they are the ones that are responsible for creating the eggs and sperms, or the final sex cells that are going to make the next progeny, that are going to make the next generation. For asexual organisms, of course, any mutation that happens is going to be transferred to the next generation. But for sexually producing organisms like ourselves, it only matters as far as the transfer to the next generation is concerned, if the mutation is in the germline or not. So here you can see a condition where you have the gametes from the male and the female, and this is from just a normal cell. So you have these zygotes that form, and they have their germline, and they are having whatever they need to do, and then they're giving rise to all the somatic cells in the parent. And then when this is a normal cell going through just its normal lifespan, when this cell creates its gametes that combine with another gamete to create another zygote, the offspring is going to look the same as well. It's not going to have any issues with mutations in this case. However, now let's say what happens when there's a mutation in either the germline cells or the somatic cells. So here in the parent, there are two mutations, one that affects the germline, which is your A, and second, mutation B, that does not affect the germline and affects the somatic cells instead. So the mutation that affects the somatic cells early on in their development, so when the zygote was formed and the organism was developing, whatever this organism is, at an early point during differentiation, at the early point during development, some of the cells got mutated, or one cell got mutated in the somatic lineages So every cell that arose from that mutated cell, every different cell type that arose from there, was also carrying this particular mutation in that resulting parent. The cells that were somatic cells that were not part of the same cell lineage as the one that was mutated were not affected, and they developed normally, and they just had the same normal wild type alleles or whatever alleles they started with. Now, in the second case, so that's your somatic cell mutation. And in the first case, you had in A, you have your germline cell that is affected. So in the zygote, as the organism develops, it creates germ cells as well, right? It creates your ovaries and your testes, and then all the germ cells that are going to give rise to eggs and sperms, and whatever your male and female counterparts are. And in this case, when the germline cells are affected, that particular mutation is going to be carried in all the germ cells that are going to be produced from that particular lineage. So once the mutation happens, all these resulting gamete cells or germline cells are going to be mutated they will all contain that mutation, and they will create gametes that contain that particular mutation. Now they will not contain mutation B because that was a somatic cell, so it never will reach the germ cell. But the mutation A will be transferred to the gametes. So we have now a mutated gamete, a gamete that contains this mutation A, and it's going to combine with in this case, a gamete that does not contain that mutation, but the resulting zygote will carry that mutation, will be a carrier of that, even if it doesn't have, even if it's a recessive phenotype, it still has that allele. And now the progeny, the offspring that comes from this particular mutation, will contain that in every single cell, both the germline as well as the somatic cells. And you will see the effect of it, if any, especially if it is a dominant phenotype. And so that is why it is important to note where the mutation is occurring. Somatic cells only affects the original organism, and it's not going to be transferred. So, you won't have to worry about it taking part in evolutionary chain or affecting the offspring, the progeny from those individuals. But a mutation that happens in the gametes or in the germline cells will affect the progeny. And that is something to remember. Now we can look at mutation rates in any organism based on experimental data. We can do these experiments in laboratory. Usually we do these experiments with simple organisms that have lots of progeny and short lifespan, so you can look at multiple generations and overall effect on a lot of different cells. So in this case, we actually use mutants to begin with to make our life easier. So typically you are going to use, and if you're using, let's say, bacteria, which is a very common model system for experiments like that. Yeast is also Saccharomyces cerevisiae, so as a eukaryotic model system is also used many times, again for the same reason, because it will multiply very quickly and it will give lots of progeny. So in this case, we typically use organisms that have a mutation that only allows organisms to proliferate or to survive under certain conditions and not all conditions. So for example, in this particular example, you have a mutant E. coli cell. So this is a bacterial cell that requires that it is provided histidine in order to proliferate, divide, and grow. It is not able to create that itself because there's a mutation in the histidine gene, so it has become inactive. That pathway is inactive. The mutation has led to the mRNA containing a premature stop codon through this point mutation, this A to T change. And that premature stop codon results in the enzyme not being made because the protein product is truncated and is never able to do its job, which is the enzyme that's required to prepare histidine. So in this case, you start off with this culture of E. coli with this histidine gene mutated so that it requires histidine in the media. And then you let them grow in this media rich with histidine, um, until they reach a certain point. As cells divide, they're going to start getting random mutations. They're going to spontaneously arise with these mutations. You can also give them certain mutagens to cause mutations to occur if you wanted. And once you have allowed them to grow and multiply a certain amount of time to allow these mutations to occur, you would then spread a sample of these cells onto petri dishes that lack histidine. So remember, we provided them histidine all along the way so they could proliferate, they could grow, they could divide, and we let them just do their job, just multiply, and as they're multiplying, spontaneous mutations are occurring because we're wanting to look at the mutation rate in that organism, right? Without any effect initially. And then we are going to spread that out onto plates that don't have histidine. So if all the cells remain the way they were, no cells will grow because remember, they require histidine to grow. But if one of them, if they weren't stable and if there were mutations occurring, one of them might have a mutation that now reverts it back to the original, leading to a colony forming. And that rare colony of cells will now have a mutation that allows that enzyme to actually be produced and to work and make histidine just like normal. And thus the organism is able to survive on this plate which didn't have any histidine in it. It's called the reversion of that original genotype. So that reversion corrects that original mutation and allows them to grow and survive in the absence of histidine. And then we can look at how often that happens to look at the mutation rate. The more colonies you find, the higher the mutation rate, because obviously more colonies were reverting back to the original phenotype. So this is something that we do regularly to look at mutation rate in specific portions of the genome. Some portions of the genome are more likely to be mutated than others, especially regions that have lots of repetitive sequences. They are going to be more likely to be mutated because it's just a more possibility of mutations in there versus more unique sequences. Smaller proteins versus larger proteins, and you can look at all of those to see what the mutation rate is. The mutation rate also can change based on the use of the gene, how much it is utilized, how much expression level it is. And so one of the things that we saw was the percentage, looking at percentage of population that is lactose intolerant globally, we could see that the mutation rate was different in different parts of the world. And there was a selective advantage to digest lactose due to point mutations in the sequences that correlated with an environmental change, which was the change as to the ability of us to domesticate cattle. And as we domesticated cattle, those areas were more likely to utilize the lactose gene and were more likely to get those mutations that would allow us to digest lactose and allow us to use that as an energy source. So when we monitor the lactase gene globally, we actually notice that different areas have different percentage of population that is lactose tolerant and is able to digest it, obviously, right? But also that it is happening not because of mutation in the lactase gene, but rather in the regulatory sequence of that gene. And there are actually two different mutations that arose, one in Europe and one in Africa, that gives rise to this ability to utilize lactose as an energy source. And so these two mutations they occurred in the same regulatory sequence, but it happened independently and then spread across the globe with the population moving from one place to another. Okay, so how can just changes in regulatory DNA sequence have such a dramatic, change at the end of the day? Well, to look at that, we're going to use an example of two species that are, we are looking at the same gene sequence. We're looking at three different genes that are all affected by the same transcriptional regulator that can, and different regulatory sequence for those three genes. So in species A, you have these three genes in a sequence on a piece of DNA that are controlled by, each one of them has their own regulatory sequence, and you have a transcription regulator that turns on gene 1, and that gene, and when it switches on gene 1, its presence switches on gene 1, it produces a product that can then bind to the regulatory sequence of gene 3 and induce the expression of gene 3, leaving gene 2 in the DNA sequence just quiet and switched off. However, in species B, you have the same transcriptional regulator that turns on gene 1, but this time gene 1's product, because of mutation, the way the mutations have happened, gene 3 is actually missing that regulatory DNA sequence that allows this transcriptional regulator, gene 1 product, to bind to it. And instead of gene 3 having that sequence, gene 2 has that sequence. So in this case, gene 2 will be activated because that's where the regulatory sequence is in this particular organism, maybe because of a switcheroo, or a movement of genes, just like we talked about earlier, right? So you can have this transposition, or you can have gene duplication and divergence. Any one of those reasons that you could end up with the regulatory sequence to be excised from one place and put into the next, or adjacent area, or even a different gene altogether, or different chromosome altogether. But in this case, in species B, you are going to have the same gene 1 product now binding to its own regulatory sequence, just like it did before, but that regulatory sequence happened to be activating gene 2 instead of gene 3, leading to a completely different product and thus a completely different organism in that case. So that's actually something that happened when we looked at several different organisms. We see exactly that happening in them. So most common reason why something like this would happen would be a gene duplication, or and then subsequent divergence of those genes or those areas. Gene duplication can happen because of issues in crossover, right? Especially if the areas have the same kind of short repeated DNA sequences, a lot of repetitive sequences that are adjacent to each other in the homologous chromosome. So here you have the homologous chromosomes, you have those genes, you have these regions. These are the short repetitive DNA sequence where you can have these issues arise. So when these chromosomes kind of try to match up for crossing over, they may cross over incorrectly by matching up with the wrong repetitive sequence. This misalignment can then lead to unequal crossover product at the end. So you can end up with a short chromosome which is missing any copy of the gene, versus a long chromosome which has these two genes tandemly, right, in tandem, so duplicating that product. And now you have these three independent DNA sequences, very repetitive sequences in there, and such simple effect can lead to a drastic change at the end of the day. So these duplications can obviously then lead to a very, very different product at the end of the day, right? And that can lead to divergence of different species, different sequences, different protein products as well. And they can give rise to related protein products, or they can give rise to very different protein products and create what we would call families of genes. Something like this has happened with the globin gene, hemoglobin, for example, right? Where multiple rounds of this duplication and divergence have led to development of several different forms of the globin gene. So you have all these different variants that are present over millions of years, as things have duplicated and diverged multiple times in that portion of chromosome 11. And now you have several different copies of this globin gene with different flavors or isoforms or alleles present within them. The original, or the ancestral globin gene encoded just a single chain of the globin molecule that gave rise to now that was just used 4 times to create that 4-chain hemoglobin protein. But in modern humans or modern mammals organism, you will see that there are these multiple isoforms, and there are that pair of genes, the alpha and the beta, that both are produced to create that final hemoglobin molecule with the 4 chains, where there are 2 alpha and 2 beta units together. The advantage of having this versus the single chain globin molecule is that the single chain globin molecule could only bind one oxygen molecule, and so obviously you would require a lot more of it to get the oxygen that you need efficiently. But through this evolution of the second globin chain and these different rounds of gene duplication and mutations, you now have this 4-chain hemoglobin molecule with the 2 alpha and the 2 beta molecules, and now you can actually end up with 4 oxygen molecules per protein, this final protein product. And so it allows for more oxygen to be transferred from one place to another in a more efficient manner. Now, this kind of thing doesn't just happen at a small level, but also just a single gene level, but rather whole genome duplications can occur as well. And many times this happens in plants, and that can lead to many of your, modern crops. Actually, the ones that you find in the supermarket are a result of whole genome duplication. So for example, apple and potato, there are 4 ends. So they have 4 copies of the genome, not just 2. Kiwi and wheat have 6, and sugarcane, strawberry have 8. So these whole genome duplication can shape the evolution of these plants or evolution of these organisms, and they can lead to a very different final product than what you started with at the beginning, when there was just the normal genome in them or the typical genome in them. Now, another thing that we talked about was exon shuffling, and how that can also be a big force in a driving force in evolution and change over time. Exon shuffling basically leads to different protein products from the same sequence or from a couple of different sequences through different combinations of exons. So different combinations of those domains, the protein domains that are present within that DNA sequence. So in this case, you are looking at epidermal growth factor with its just one simple domain, and then you have these 4 different proteins that have other domains as well. So you have epidermal growth factor, chymotrypsin, and urokinase. Urokinase and epidermal growth factor both contain this one domain for growth, and then you have chymotrypsin and urokinase also share a domain between them as well. And so through splicing, incorrect splicing, or alternative splicing of these gene products or genes as they are transcribed gene transcript as they are turning into mature RNA, you can lead to different combinations of these proteins, right? You can completely change the biology of the protein. You can completely change the final effect because different domains are going to be combined together. So, for example, in factor VIII, you have two of the green domains, and then you have an alternative domain. In plasminogen, you have several of the domains that you are also finding in urokinase kind of spliced together as. So it is actually theorized, or it's actually proposed that our human genome, which contains about 24,000 different proteins at minimum, happened from duplications and shuffling of just a handful of these domains that have been spliced together in different orientations and um, different combinations. And each one of these protein domains would be anywhere from like 30 to 50 or a few amino acids long. And this can happen. The reason for that is that this whole splicing process um, that happens, it happens through recombination events, through cutting and pasting at particular sequences, and there's a lot of flexibility in that because it's the same sequence essentially, with some flexibility that is getting cut in each of these exon and intron junctions, and so it's possible to splice these different things together in different ways. And because of that, you end up with all this diversity of protein structures and function and the way they behave inside the body, with just essentially small parts, right? These are just like small combination of parts. It's a handful of parts, and you can combine them together in any which way. Um, kind of like circuit building, right? Where you have the same different parts, but they can be combined together to do different functions, completely different functions at the end of the day. So again, an extremely important driving force for something like this. Another driving force, again, a major one, is these mobile genetic elements. Now, mobile genetic elements or jumping genes can induce many changes in the organism, both at the sequence level and also at the phenotypic level, because they can disrupt both the regulation of the gene or the presence or absence of the protein product from that gene. So here in this particular example, there's using Drosophila melanogaster, because of the jumping gene in their mobile genetic element in there, this normal fruit fly, which is has a normal body plan, a typical body plan, can be made to have progeny, end up with progeny if the mutation is just right, that replaces the antennas with legs, just because the regulatory DNA sequence for leg formation got activated when the antenna was supposed to be, due to movement of the um, mobile genetic elements or the transposon that was present there. Now, this can obviously cause some deleterious effects, but it can also be helpful in some cases. Insertion of particular mobile element, genetic element, allowed us to duplicate certain portions of the genes and of the genome in the original corn plant to give us these really plump seed, modern corn, as opposed to an ear of corn from the original corn plant or teosint, which looked very different and did not have the same plump, juicy seeds like the modern corn does. So now we learn a lot more about this in the next chapter when we talked about how transposition happens. It's also something we've talked about already in the genetic lectures, and I'll link that at the end as well. Now, the final one that we'll talk about is horizontal gene transfer. Again, a very important driving force for gene evolution, for evolution of organisms. In this case, this is when we think about bacteria, we think about asexual reproduction where they're just dividing by binary fission. But there are sexual ways that and there is sexual reproduction of sorts in bacterium as well. Certain types of bacteria can produce sexually through the presence of their sex pilus. These particular organisms can exchange genes from one to another using this sex pilus. And usually what they will transfer would be either the entire genome or small portions of it to the other organism to convert it from one phenotype to the other. And this process is called horizontal gene transfer when that is able to happen. We will learn a lot more about that in the coming lectures as well. So the way we generate genetic variation, again, just to kind of recap, how are some ways that you can do that? In sexually reproducing organism, one major way to do it is to change the germline cells so that the change or mutation can pass on to the progeny. Again, somatic cells mutations will not get transferred to the progeny and will not have that effect. These point mutations can cause obviously failure of the actual gene sequence or failure, changes in the actual gene sequence, but they can also create mutations that are going to lead to disruption of the normal DNA regulation for that particular gene or for copying and repairing DNA related to that gene. Mutations can also change the ability of these cells or ability of the genome to function properly through that as well. You can also have DNA duplications because of issues with crossover, for example, that can give rise to families of related genes through multiple rounds of duplication, especially, and multiple rounds of this duplications and subsequent divergence of the sequence can produce these really variable alleles or genes that are all part of the same family, and that can give rise to new phenotypes over time. A whole genome duplication has also been a big driving force in evolutionary history of many species and is especially used in agriculture and crops. Okay, so next part we are going to be talking about how we can reconstruct family trees looking at the mutations and their sequence over time in different ways. So when we examine our phylogenetic tree, we can look at look at all the genome sequences that we have up till now. We can look at relationships between the modern life that is present on Earth today and match it with different organisms that have survived at different times in history. And using that kind of reasoning, we can look at the last common ancestor between different groups of organisms on that family tree, and then also at the last common ancestor for all the organisms that are present within that portion of the family tree. So for example, here in this case, we are looking at genome of human chimps and gorillas, and orangutans. And we are looking at how divergent they are from each other over time in millions of years, and based on the number of sequences that are different, number of nucleotide changes that have happened, percent change in that nucleotide sequence during that time frame. And so you can see how, both chimpanzees and gorillas are more closely related to humans compared to orangutan. And they have both diverged from each other as well over time. And you can find that the last common ancestor of human and chimps was a lot more recent than orangutans, for example, which was a lot more time ago, that nearly 15 million years ago versus only 5 million years ago. And looking at the DNA sequence changes, you can see that this is just a little bit over 0.5% different in the DNA sequence. Um, Gorillas, chimps, and human, when you look at all three of them, they're about, still less than 1% difference. And even when you go to orangutan over this 15 million years, at the end of the day, there's only 1.5% difference. That's very little to give a very big impact. So you can see how even small, tiny changes over time can have a big phenotypic effect due to the divergence of genomes as they are making those changes over time. So here you can actually look at a sequence of your genome. So in this one, this is looking at a particular protein, which is the leptin, a mature leptin protein, which is a small protein to begin with, but we are looking at about 300 nucleotides over 5 contiguous segments, so 5 segments over this sequence. It's taking care of about 300 amino acids, or 300 nucleotides rather. And this is the sequence, and what we're looking at is human and chimp DNA primarily in over these 5 rows. And you can see that there are very few differences in this 300 nucleotide sequence. You actually have only 5 nucleotide differences between chimps and humans. Some of them are also changed in gorilla. Some of them are the same in gorilla as humans and are unchanged in them. So you can see that here, all these sequences that are present here, you can see that this particular gorilla sequence is either is matching the chimp hair, it's matching the chimp hair as well. It's the same as human hair, or human's hair. It is the same as human's hair. So there are very, very few differences in the sequence. The last 145 nucleotides that are after this are identical in both. So there's no difference, zero difference between chimps and human for that particular sequence. So these are, and when we look at this further, we also realize that out of these 5 changes, only one results in an actual change in amino acid sequence. The rest of it, you're still getting the same exact amino acid as you were getting before. So you can see that at each point, this is still a Q, this is still a K, right? This is still a lysine, still a proline. Here, it's changing from a valine to a methionine, but everything else is still the same in this particular sequence. So you can see how closely related these present day species are to each other looking at a sequence like this. Now, how do these, again, changes happen was through the presence of those, different forces that were acting on the sequence, whether it was through the environment or mobile genetic elements or through point mutation just because of the mutation rate, any of those things. So another driving force, as we talked about, was the presence of mobile genetic elements. And actually, that's one way that you can fast forward evolution, right? Or these changes, because they have the ability to transpose from one place to another and jump and obviously take along some of the genome or disrupt genome. And the more they do it, the more issues can arise or more changes can arise. And that can obviously drive that evolution a little bit more. Here we are looking at human beta globin gene cluster and mouse beta globin gene cluster from that chromosome 11 like we saw before. And you can see that in this particular portion of the genome, you have 5 different globin genes in this gene cluster in humans, while in mouse you only have 4. And those 4 obviously have changes as well, right? So each one of them are a little bit different. And this is one way that you can see how even in this time that the mouse and humans have diverged from each other, they have diverged so greatly that you do have different isoforms of the genes and you have different number of these genes in that gene cluster. That shows how long the evolutionary time might have been between those two species. Another thing you will notice is that you have these different shapes and the colors in there, the green and the red. The green are the allo sequences, which is again a repetitive sequence that can act as mobile genetic elements. And these are going to specify presence of mobile genetic elements. Now the type, the sequence, and the position of these genetic elements is different in the human versus the mouse. Both have some allo sequences and both have some L1 sequences, which both act as positions of these mobile genetic elements, but they are of different sequences. And they are in different amounts in each, right? So this tells us that those two clusters are arising independently of each other and are inserting in these spaces independent of each other. So they may not be derived from the same original place. Now, having said that, there is some similarity between them. In both cases, while the positions are slightly different, they are avoiding any of these actual gene sequences. So they could be because of natural selection kind of taking care of the ones that end up in there, because that disrupts the gene function, so it dies out over time. But definitely these mobile genetic elements are mostly present in those regulatory sequences, or not even regulatory sequence, on those interspersing sequences and not in the actual gene space that is present in there. So in spite of the gene shuffling, we can still recognize the basic block structure between the two, and we can recognize that same sequence of events happening in those two spaces. This is actually also called conserved synteny. So here, again, looking at that accumulated mutations that have happened in a particular portion of genome between human and mouse. You see an exon intron junction in here present as well. And you can see that if you look at the exon of your genome, of this particular portion, right? This is the genome, and you look at the mouse versus human, there are very few differences between these two sequences, right? There's only 1, 2, 3, 4, a handful of sequence changes. However, if you look at the intronic sequence, it's all over the place. It's very hard to even say that they're from the same place. So the intronic sequence, because it wasn't part of that gene, at the end of the day, the final protein product, it didn't play a role in it, ended up being a lot more divergent and had a lot more mutations accumulate in there, because they didn't really matter in the big scheme of things, while the exon sequence is preserved and conserved a lot more closer, a lot better, to make sure that those sequences are there to have the final protein product you need to do the work that you want. Again, looking at looking at a small region just like that across multiple organisms, you in fact see that happen every single time. So here you have two really large intronic sequences and this one exon. And you notice that, obviously in chimpanzee, orangutan, there's a lot of similarity because those are very closely related modern sequences. But across from the fugu fish to chicken to possum, mouse, dog, cat, all the way up to chimpanzee, and looking ahead at the human genome, you find that this exon right here is conserved all the way through, while a lot more changes have occurred in those intronic sequences. A lot more mutations have happened, and obviously the more far back the the more loosely that organism is related to us, the more far back in the evolutionary tree that organism is, the less identity you find in that region. But still, the exon, the actual exon for that particular protein product, still is conserved all across the genome, all across the species a lot better and to a high degree. And again, you can see some other conserved intron sequences as well. And that could be that maybe they play a part in some regulation for another gene. And they may contain some regulatory sequences of their own in that area that make it important for that area to be conserved. This can give us clues as to which parts of the genome we should be worried about, or that we should be focusing on for looking for the function or structure and things like that, because those are the regions that are conserved are the ones that are going to be important for that organism at the end of the day. Now, regardless of this, some genetic information, some particular sequences are so critical that they are conserved all through the life forms. So from simple bacteria all the way to humans, everything in between. So here you have an example of something like that in the shape of the gene for a small ribosomal subunit RNA, which is conserved from beginning of life all the way through now. You see that even comparing to E. coli or Methanococcus and looking at the human sequence comparison between them, most of the genomes are conserved. Most of that gene is conserved. Most of the nucleotide sequence is conserved. There's this one place in E. coli where there is a nucleotide that has either been deleted or something has happened differently there, but outside of it, everything else, you see it's like over 90% conserved in both species compared to humans as well. This again lets you know the importance of that particular protein, that particular gene, and its function in our life because of its conservance all throughout life forms. So looking at all these sequences and the conservation of specific sequences across genomes, across organisms, across life forms, we are able to create this tree of life that shows the three main domains, which are your bacteria, archaea, and eukaryotes. And within, again, all arising from the same common ancestral cell at some level, at some point, millions of years ago, but then diverging over time through these mutations, duplications, transpositions, all kinds of events that happen in order for evolution to occur and in order for changes to occur and to adapt to the environment, to survive in a space, and lead to this big diversity that we see today in those life forms. Now, looking at these, majority of these organisms are actually pretty small, and you cannot see them with your unaided eye without a microscope or something like that. However, these few tiny little space, these human yeast maze, right, plants, a small portion of your eukaryotes is all you see visibly, which are your animal, plants, fungi. They represent a very tiny subset of the diversity of life that is present out there. So there's a lot more out there than what we can see and what we can examine any given time. So just recapping this portion of the lecture, to reconstruct life's family tree, we take advantage of these changes that have happened over time in our genomes, both in the important exon protein products as well as in those intronic or regulatory sequences that are there. We can look at genetic changes that provide selective advantage to an organism. We find that those are more likely to be preserved and conserved. Organisms that are more closely related to each other will have genomes that are going to be more similar to each other in organization as well as their sequences. Organisms that are going to be more distant on the evolutionary tree, that are more different from each other, will have less similarities, although there'll still be large portions that will be conserved because of their importance in the function and structure of those organisms or just life itself. Genomes, functionally important genome regions will show up as islands of conserved DNA across all organisms or across a large portion of life forms. and that is important to understand which areas are important for us to study further. And genome comparisons can show that a lot of these genomes can gain and lose DNA relatively quickly based on the way the nucleotides are changing and the percentages that are happening there. And finally, we can look at the sequences that are conserved, and we can look at aligning them within each other to trace even their most common long ago evolutionary relationships, even when you can't physically see any relationship between them, just like you can't see really a difference, but any relationship between a simple E. coli compared to humans, but yet, if we look at it at a DNA level, we find that there is still a lot of sequence conservation and presence of the same genes and protein products that are needed for survival in both cases. Now, next time, I'm going to start from here and we'll talk more about how mobile genetic elements and viruses can help further this evolution or fast forward this evolutionary change and help to evolve life forms into more intricate things. Hope this was helpful. Take care. Bye-bye.